Welcome to the small symposium of the 3R Stimulus Fund of Utrecht University, the University Medical Center Utrecht, and the Utrecht Animal Welfare Body. My name is Monique Janssens. I'm the communications consultant of the Animal Welfare Body Utrecht. I want to ask you please to turn off your camera, mute your sound until the moment for questions so that we have optimal sound and vision. We have a small group today, but we will make a recording to make it available for anyone to see later. After each presentation, there will be some time for questions. I don't think it's necessary to use the chat. You can just unmute yourself, put your camera on and start talking. Please keep your questions short. We will have two presentations today. The first presentation is by Martijn Evers of the University Medical Center. Please, Martijn, you can start sharing your screen and I will announce your topic. Martijn will talk about getting nanoparticles in the right place in mice, but now this is possible using fewer mice than before. Martijn, please go ahead.
Well, thanks. First of all, thanks for your interest in this presentation. Today, the topic of my talk will be how to test more nanoparticles with fewer mice. I'm a PhD student at the Central Diagnostic Laboratory, part of the nanomedicines group, and we're actually interested to deliver RNA therapeutics via nanoparticles. First, I'll give you a short introduction about RNA therapeutics and what lipid nanoparticles are. And then I'll tell something more about how to use DNA barcodes to test more nanoparticles in fewer mice. Well, RNA therapeutics, they act mostly on the level of proteins. So as you might know, in our body, a gene is transcribed to an mRNA transcript. And that mRNA transcript is translated into a protein. And these proteins are essential to our body. Flaws in these proteins, misfolding or an increased amount of a specific protein or the absence of a protein, they can lead to disease. And therefore, therapeutics such as RNA therapeutics, they can address many diseases at the level of DNA/mRNA. For instance, they can increase the expression of a protein simply by administering mRNA, or you can reduce protein expression via a mechanism called RNA interference. You can break down the specific mRNA transcript. Well, more recently, they discovered also a tool from bacteria, CRISPR-Cas9, which enables us to make small changes to the gene so that we can, if there is a small error in a gene which leads to a false protein, you can correct for that. And that's all being done by small or large RNA molecules. So these molecules are very potent. However, there are many barriers for the successful development of such RNA therapeutics. There is no such thing as a RNA therapeutic tablet, which you can easily swallow. You have to inject these RNA therapeutics directly into your bloodstream. But even then, if you directly inject those therapeutics into the bloodstream, they are either rapidly excreted via the kidney or immune cells. They eat those RNA therapeutics. Or in your blood, there are also nucleases, and they actually break down these RNA therapeutics. And let's say that a RNA therapeutic overcomes all these barriers, then their size and their charge, they limit cellular uptake. And these therapeutics, they are active in the inner part of the cell, in the cytosol. So RNA therapeutics are very potent in principle. However, it is very difficult to translate that into a successful therapeutic. And that's where our field of interest is, namely nanoparticles as a solution for all the barriers I just discussed. if you try to load your RNA therapeutic in a particle, then that particle might prevent the RNA therapeutic from being excreted. It also protects it from nucleases. And at the same time, as you can see in the picture on the left, it also provides a vehicle or something for the cell to take up the RNA therapeutic and get it in the place where it needs to be active, the cytosol. And of course, there is no such thing as a nanoparticle. There are many different types of nanoparticles. You can make particles of lipids, you can make particles of polymers, you can also make a sort of conjugate to some other sort of chemical modification to your RNA therapeutic. And our lab, and what we're talking about today, is about lipid nanoparticles. So these are small particles made of lipids. So these particles, as I just said, yeah, lipid nanoparticles are made of lipids. However, it's a bit more complex than that. So if you look to the left lower quadrant of my slide, you see a schematic representation of how such a nanoparticle looks like. And you see it's made of several different categories of lipids. You have a PEGylated lipid, a PEG-Lipid. You have cationic lipid. These are positive lipids. And you have helper lipids. And since it more or less is comparable to a lipid membrane, there's also cholesterol in it. However, there's no such thing as only one cationic lipid or only one PEG-Lipid. And that's what you can see on the right side. In the right upper quadrant of the slide, you see 3 different cationic lipids. And if you then look to the left side of these lipids, you see the lipid tail. And on first notice, you might not see that many differences between these lipids, but there are differences. For instance, the lipid DOTAP, it has only 1 double bond in each lipid tail, so 2 in total. The other lipids, D-lin KC2-DMA and D-lin MC3-DMA, they have 2 double bonds in each lipid tail, so 4 in total. And if you then take a closer look at these lipids, you see all kinds of differences in their chemical structure. And it is important to know that these small differences in the lipid structure can have an influence on the functionality of such a particle. The same on the right lower quadrant, you see different PEG-lipids. There you see also, again, differences at the left side of the lipid. So the lipid tail length varies. The upper lipid has a C14 chain, so 14 carbon atoms. And the lower lipid has 18, so they differ in length. And it's important to know that these differences in lipids can have functional influences. At the same time, you can also understand that the ratio in which you use the PEG-lipid, the cationic lipid, or the helper lipid can also have influences on its functionality. So the recipe, how you make your particle, has a big influence on how the particle functions. And you can make a lot of recipes. And here I just want to show 2 examples about the influence of this recipe on the functionality of such a particle for RNA therapeutics. So the first example is about the PEG-lipid. And the PEG-lipid influences or can influence the pharmacokinetics of such a lipid nanoparticle. Again, on the left part of the slide, you see 3 different PEG-lipids. These are the same as I showed in the previous slide. These lipids, they differ in their lipid tail length, so 14 carbon atoms, 16 carbon atoms, or 18 carbon atoms. And if you incorporate these lipids in a particle, so in one particle only has C14 PEG, another particle has C16 PEG, and another particle has C18 PEG. And you test these particles in animals, so you directly inject them in the bloodstream of a mice, you see that their behavior is different. So particles with a C14 PEG-Lipid, they are rapidly eliminated from the blood, as you can see in the right graph, and then the left upper part of that graph. Whereas particles with a C16 PEG-Lipid and a C18 PEG-Lipid, they are excreted slower. And that's what you can see in the right part of the slide and then the left upper graph. So for C14 particles, the percentage of the injected dose, which is in the blood, more rapidly decreases compared to C16 and C18. And that can have functional influences. And here they only tested 3 different nanoparticles. If they then wanted to optimize the molar compositions, the recipe of your particles, so to say, and looked for their transfection efficacy. Well, transfection efficacy is a term which describes the ability of a particle to get the RNA in the cytosol. Then they saw that the ratio of these different lipid components, if you change that, then that has influence on the ability of the particle to get the RNA therapeutic at the right place in the cytosol of the cell. And that's what you can see in the left lower graph. There you see on the y-axis serum EPO, and on the x-axis you see 33 different nanoparticles. So what they did is they made 33 different lipid nanoparticles. They put EPO mRNA in all of these particles. So the mRNA for the protein EPO, they put that into the particle. They administered these particles to mice. And then after a certain period of time, they looked if they could detect EPO protein in the blood of these mice. And the better the particle is at delivering the RNA therapeutic, the higher you would expect the protein concentration to be. And what you can see from this graph is that the recipe of your particles, so the constituents, so how much cationic lipid you put in this particle, how much helper lipid you put in this particle, how much cholesterol you put in the particle, and how much PEG-Lipid you put in the particle, and the ratio between it, that influences the ability of the particle to get the RNA therapeutic at the right place. And you see that that could differ between, let's say, their original formulation. They optimized that by almost 9-fold just by changing the recipe of the lipid nanoparticle. But you could see that that graph on the bottom of the slide contained 33 different nanoparticles. That means that in that study, they already used 99 mice just to test this. And if you know that the correlation between what you test in vitro is very low to what we actually see in vivo, that means that we actually want to test most nanoparticles in vivo because there is no predictive value or hardly any predictive value in what we see in vitro. So if we want to optimize our particles, that normally requires testing particles in a lot of animals. So then is the question, what if we could test the critical attributes of these different lipid nanoparticles, but not use 99 animals for it, or 90 animals, but only a couple, or even in a single animal? And that's what the group of James Dahlman already thought of about. And this might get a bit technical, but what they did is they used DNA barcodes for high throughput testing of lipid nanoparticles. And what they did is they developed a DNA barcode. So that's a unique piece of DNA with its own sequence, and they put in every different lipid nanoparticle a unique barcode. And what they then did, they pooled these particles, so they mixed these particles, and they administered it to a single animal. So, as an analogy I've come up with is you have to compare it to the supermarket. So if you take a lot of product, each product has its own barcode. And if you then go to the cash desk, they scan your products and they know which products you had and how much of the product you had. And this technique actually does the same. So each different particle has its own barcode. Then if you mix them and you administer them to mice, and for instance, you take several organs and you go with those organs to the cash desk and start scanning all these organs for the different barcodes, then eventually you would know which particle do I find and how much of that particle do I find. And that enables us to make comparisons between these particles and then select for the characteristics we would like to have. Though the scanning we perform is a bit more complicated because that is done using next generation sequences. And then what we do is, based on these data, we know how much of a barcode do we have and which barcodes do we have, and that we can then use, because we know which particle has which barcode, to compare these particles. So the main technique was already developed, but for us it was interesting to see if we could apply this technique within our infrastructure and apply this technique to our type of particles. And that's why we wrote the proposal for the 3R Stimulus Fund. So currently we test in our lab particles in a lot of animals. But what if we test our particles using this DNA barcoding approach? And we were interested if we then, we have already results of our particles. If we then use this new technique, can we see, or do we get the same results? And we actually did that using those PEG-Lipids, because we know that these PEG-Lipids have a certain profile in animals. You could see that C14 particles containing C14 PEG, they are more rapidly eliminated compared to particles containing C16 PEG compared to particles containing C18 PEG. So what we did, first we did not use animals. We started with cells in a small well or petri dish. That's what you can see. That's the pink wells. And what we did is we administered particles individually and we administered particles mixed. And what we expect, also the uptake in cells, is influenced by this PEG-lipid. We know that particles containing C14 PEG, they are more rapidly taken up by cells than particles containing C16 PEG. And those are then quicker being taken up than particles containing C18 PEG. And if we then analyze these data, we could see that the trend was similar, both if we administer individual particles and if we mixed our particles. And that is what you can see on the y-axis, the relative occurrence of the barcode in the cell, and now for the different particles. So the trend in individual particles was the same as when we mixed our particles. Then we continued to animals. So we administered our mixed nanoparticles into an animal, into a mice in this case. And we took some blood draws, and then after 4 hours we took the organs of the animal. And since we already know, based on historical data, what the pharmacokinetic profile of these particles is, we can compare the data we have using our barcoded nanoparticle approach to these historical data. Fortunately, I cannot show you these data yet because they're still under analysis, but this is the way we're heading, because we eventually want to use this technique to screen multiple formulations, over 60 different formulations, for their pharmacokinetic profile after myocardial infarction. And that's what the future is about for us. And instead of using almost 200 animals, such studies would then only require approximately 4 animals. So you really efficiently reduce the use of animals for these type of studies. To conclude, if this pilot, which we're still analyzing, is successful, then we have a tool to test many different types of nanoparticles in a single animal. I hope it was a bit clear. Thanks for your interest. And if you have any questions, you can ask them right now.
Thank you very much, Martijn, for this extremely clear explanation of something quite difficult to understand, for me at least. I can see there's a question already from Wim. Wim, please unmute yourself and you can ask your question.
Yes, hello, Martijn. Thank you very much for your presentation. I do have a question. Do you think this technology is also of interest for RNA vaccinology?
For RNA vaccines, you mean?
Yes.
Yes, of course. What I think you can do in RNA vaccinology, you have, I guess you talk about administering mRNA, encoding an antigen.
Yes.
And yeah, so what they've did is they've tried to combine this method to reporter animals of which are sensitive to mRNA. So these animal models, they express a cassette, which is a flocked stop cassette, limiting the expression of TDT tomato. And if you then administer recombinase mRNA, the cell turns red. And what they did, they combined such reporter systems with the barcode system to see in which cell you can successfully deliver mRNA. And then they can select for the right particles. So, for instance, if you know which type of immune cell you want to target for RNA vaccinology, like dendritic cells or macrophages, yes, you can optimize your particle using such systems for RNA vaccination. I think you can use them, yeah.
Yeah, and then a small other question. So you decided to stick to the animal model. It gives better results.
Yeah.
And with this method, you need far less animals than in the past. But how predictive are the kinetic profiles that you find in mice compared to the profiles in men?
Ooh, that's difficult. That's there we reached boundaries of my knowledge. For nanoparticles, I do not necessarily know that correlation. But in general, we know that still mice, the data in mice, they sometimes do not correlate well with men. But that's only a general comment. For nanoparticles, I don't know.
Okay, thank you.
Okay, the next question is from Jan.
Hi Martijn, Jan van der Valk speaking. Thanks for a very clear talk, it was really good. I have 2 questions. First one is a bit related to what Wim asked you before. There is a low in vitro to in vivo correlation. I wonder what part is so low correlated? Is it only the the excretions, so the kinetics, or also the cellular uptake that is difficult to correlate?
I think it's a combination. What we normally do is we just look at the functional data. So, for instance, if we want to do an RNA therapeutic which effectively silence a protein, and we do that in a 96 well plate and then compare it to animals, you see that the efficacy, that there is no good correlation in that, and that those differences can occur at any stage. A petri dish is simply too much an oversimplification of the complex, the complexity of an animal. So the nanoparticle has to circulate, then has to be taken up by the cell. If it's taken up by the cell, then it has to escape from the endosomal compartment before it is in the cytosol. And each of these stages, there might be differences.
Sure, I can imagine that. But can't you use the in vitro system as a pre-screen to see in the first instance whether the DNA actually enters the cell and then has some kind of effect? Because if it doesn't enter the cell, you don't have to test it in vivo. Yes,
I fully agree with that reasoning. But as you can see here in this figure, lower part left side, you see that more or less this method was developed as a solution for that process, or as an alternative, because current in vitro models were used, and then you only select, so to say, an amount of finalists, and then you test them for in vivo. But what they also did using this technique, they tried to test over 2,000 nanoparticles, I believe, in vitro and in vivo, and they could hardly find any correlation. So
it's interesting, though, isn't it?
That's really interesting. So, yeah, that's why they tried to also go to more these kind of systems, because they ran into issues using the old method.
Right. Well, my second question relates to the mixing. I would expect at some point that there is also interference between the different DNA particles, but you didn't see any interference. What does that mean? That they all act through different pathways that have no interaction with each other?
I don't think that is necessarily the case, that we're talking about different pathways. So we mix them, and of course there might be competition between the particles.
Right.
So the dose at which you administer your particles is then relevant. At the same time, I think it's more of a challenge that you make sure that you're sure about if the particles itself don't mix or fuse or aggregate, and they remain stable. And that's all something you have to watch out for.
Okay, thanks.
Thank you. Kirsten has one last question, and then we move on to Claudia's presentation.
Yes, thank you, Martijn, for your presentation. I have a question about, so I understand that with the barcoding, you can distinguish between different particles that have different compositions, but I guess you also have to test like different dosages, for example, right?
Yes.
And this, I guess you cannot minimize the amount of animals you need for that with the barcoding, or can you?
If you would have, I guess it's best not to minimize for those control experiments the amount of mice you use. Because you might find a solution. I'm not sure about that, but no, I don't think you should minimize on that. No, I have no clear answer to that at the moment.
Okay, because you said on your last slide that you can go from 200 animals to 4.
Yeah.
Does that mean that you usually test 50 different particle compositions?
You would like to test as many, and at the moment we're testing 30 formulations at max in animals. But we do that the last couple of years on a quite regular basis. So there, yeah, you can reduce the amount of animals using such methods.
And have you already gone that high to test 30 different ones now with the barcoding in 1 mouse?
We're still at the moment now at our pilot study where we mix 3. We're analyzing those data, and then indeed in the coming months we are planning on testing over 30 nanoparticles. Definitely, yes.
Okay, thank you.
Thanks.
Well, Martijn, thank you once again for this great talk. We will now go to Claudia Wolschrijn. Claudia has a bit of a difficulty with her internet, and that is why we will be sharing her presentation from our office. Liedewij will help me with that. So please, Liedewij, share the presentation, and then Claudia will talk about, well, it's on now, plastinates and how to plastinate sacs of birds. Please go ahead, Claudia.
Yeah, thank you very much. Thank you for sharing my presentation. I'm here at the more or less dead end of the internet, and they promised that we would get glass fiber, and they were very happy to announce that we would get that in 2 years' time. So I hope that this crisis is then came to an end, and then we can do it all well more real life again. This is going to be a real different type of story. This is about birds, this is about teaching, and it's about air and how can we make sure that we see air, or the let's call the sacs, the containers where the air is in the bird. How can we show that to our students so that they can actually get a grip on the 3D anatomy of birds? I may have the next slide. And so if I would start with the goal of the project and what we asked the funding for, that is a way of showing and in a sustainable way the anatomy of the air sac. And I can show you some things later on how we do that until now. But the thing is that the air sacs are really thin, and you can really, they are transparent, so you can look through it, you can see other organs by looking into them. And that is what we would like to show to the students, that they know how to use them and how they can use them later on in practice. As this is for the veterinary school, so this is for veterinary medicine. And but also we use this for biomedical sciences, as some of them might even go further on with birds. And at the moment we do that by offering dissection classes. So for students dissect 1 bird in 1 afternoon, 1 chicken I should say in 1 afternoon, and in that way they get 1 chance to actually see the 3D anatomy of that bird in total. The next slide. Maybe you see the next slide already, but my internet is so slow. Yeah, there, I see it now. Okay, I said something about air sacs, and maybe if you're not so familiar with bird anatomy, then you might wonder, what are those? If you know horses, they also have air sacs, but it's a little bit different. How is that in birds? Well, if you look at the bird anatomy in this way, and you look at the picture, then you might wonder, okay, what I'm looking at? Well, you're looking at a very potent male chicken, and the white objects just in the middle of this specimen are his 2 testicles. And just on top of those you see this two pinkish organs. Those are the lungs. And just underneath it, that dark brown ones, those are the kidneys. And if you're eating chicken once every while, then you might see them as remnants also in the carcass that is left over. By showing this, and well, you don't see wings, you don't see legs or whatever, but on top is the head, and down would have been the tail. You can see that the abdominal organ, in this case the testicles and the kidneys, are immediately adjacent to the lungs, and there is nothing removed there in between. So that means that there is no diaphragm. There is no real division, or no division at all, between the thorax and the abdomen. And those lungs that you see, so the uppermost pinkish structures, they are also different in structure from what we are looking at in mammals. You'll remember, okay, when breathing, your diaphragm goes down, you expand your thorax. Within that, you also expand your lungs, and that is possible because at the end there are all these kind of alveoli, these small balloons that you can blow up a little or get blown up a little, and at that area the oxygenation of the blood will take place, so the real respiration will take place. Well, in birds it's different. Those lungs are rigid, so you can't blow them up. There is no diaphragm that will go down or backwards on 4 feet. But there has to be a kind of capacity for storage of air, and in that capacity is been provided by the air sac. So that bird is fully filled with air, and which is in these kind of air sacs, containers with very thin transparent walls, and which has another advantage because it makes the bird relatively light, and it gives them the ability to fly, as opposed to us or to other mammals. The next one, please. It's not coming in at my end here.
We see it, Claudia. So
you see it. Okay, then I take my
yeah.
Okay, then I take my own slide here. What you see here on this slide is a drawing of how these air sacs are located within the bird's body, and how the bird is breathing with them. Let's see if I can see the slide already. Yeah, okay. So at the left side, you see these blue balloons. These are the air sacs, and at the left side of it is the cranial part, so it's the head part, and the large balloon on the right, that is what we call the abdominal air sac, although they don't have an abdomen really, but it's still called the abdominal air sac. And that's the caudal part, so that's the tail part, and the pinkish part on top, those are the lungs, or it's a depiction of the lungs. And in the grey birds at the right side of the slide, you see that there is a continuous airflow going through the bird itself. So the air goes into the trachea, and then it goes immediately backwards towards the abdominal air sac, and from there it goes through the lungs. It will be the deoxygenated air will be stored in the cranial air sac, so in the upper air sac in this case, and then it will leave the body again. So there is a continuous airflow through the body of birds, making it possible that they extract oxygen from the air continuously. So during inhalation and exhalation, if you look at the airflow through the trachea. Okay, next slide, please. And I just look at my screen what that is to prevent the delay. And oh, I see it already. And as you look at that picture on the right again, what you see there, and it's pity that I don't have a mouse, but you see this lucid, transparent, tissue, and there is a kind of vessel on top of that. That is part of an air sac, and you see that it's connected to what you might remember as how do you call that in English? Kip fillet, breast fillet. So that is one of the flight muscles, and the air sac is there exactly. Thank you. Is connected to the breastbone. And if you want to study the organs of the bird, you have to remove the breastbone, because you see that it occupies a lot of space, and you have to remove the flight muscles. But then also you break down immediately the construction of that air sac, which is attached to it. So you have only one chance, basically, to look at this uppermost caudal air sac in this way. If you open up, you can see through, and then you see the lungs really lying at its back. Now, these air sacs, as I showed you on the drawing, are in the total of the body, in the whole body. They form a kind of septa, although they're really thin, but still they form compartments, or they create compartments within the body. And for veterinarians, they are important, at least if you're working with birds, because you can use them as entrances for endoscopy. If you can sex a bird, if there is at the outside no visibility of male or female phenotypes. You can also do it with DNA, but you can also do endoscopy and look at the organs, as you saw that the testicles were also within the body. And but you can also look and do an endoscopy to see whether or not there are molds like aspergillus or other deformities that shouldn't be there. So the problem is we do chicken dissection, 4 students, 1 chicken. And especially this one, this air sac, you only have one chance to see it. And if they are moving too quickly and don't really realize what air sacs look like, they have already destroyed it before they actually seen it. Next slide, please. So what do? We try to visualize them, because it's not just, and you can visualize them by casting them, and that's what you see in this slide. Here you see yellow latex, and that's been poured in the trachea, and then you have the whole of the respiratory system of a bird. So on the left side, you see these small rings. That's the cranial part, so the head part of the bird, which is the trachea. Then all the smooth areas are part of the air sacs. The stick that is sticking out to the top, that is a part of the air sac that goes into the humerus, so that goes into the wing, and that's why a bird with a broken wing might be in respiratory distress, because the air is escaping through its wing. And the light yellow, whitish part on top, those are the lungs, so the rigid part, which you can't blow up, and where the bird gets its oxygen from. So this is one way to show the air sacs, but then lose the relationship with the organs. Yeah, you see the lungs, but that's the only relationship you can see. You can't see where the heart would have been, or where the genital system is, and etc., and how you can really approach them through the air sacs, for instance. And of course there are pictures, but we all know that it's really difficult going from a 2D picture into a 3D inside of a system. So most of the students need help in transforming 2 to 3D. Of course there are texts, and And we do that with dissection classes. And as I said, we do that for veterinary medicine. That's the VS, veterinary studies. We have 225 students. For us, it's the first year practical, so we still have 225 students, and there is 1 chicken available for 4 students. We also do it for biomedical sciences, and then there are approximately 175 students, and also they dissect 1 chicken by 4 students as well. And what we see is, well, there is only 1 chance. So there are quite some groups that don't really see it. May I have the next slide? Because they missed it, and we have to do that, to demonstrate it. And and we want, yeah, the question is then, is it really worthwhile to get this chicken for teaching? What does it really bring? Does it really meet the learning outcomes that we set beforehand? Do they really, students really understand how this 3D inside is, etc. So what we wanted to do, and that's why we asked funding for, is is there a different way of preserving those air sacs, and in such a way that they are still more or less intact in the bird, if possible. And if they are intact in the bird, can we make them transparent so that they can look through them and see what other organs are behind them, on top of them, etc. And that's why that's asked the funding for. It's a kind of pilot. It was a kind of pilot study. Okay, what can we think of how we do it? We can think of filling them, and we can filling them with formaldehyde, so that we start the fixation process, and by filling them up, we keep the volume, and by keeping the volume, we might, after plastination, also keep the volume, and then there is this chance that we might, with the normal plastination, have this transparency again. What we also tried is just filling them with formaldehyde and keep wet, and also give them wet on the table, and after that would also have been a possibility, but that is not so sustainable in a way that we really want to get rid of the formaldehyde during anatomy classes as much as possible. So if you go for a new solution, and then at least in the end product, formaldehyde free. You can also think of that on beforehand. We fill the air sacs with air. You might also fill them with glycerin. We could, the latex you saw. We also filled the bird with the latex and kept what the casting is that you then afterwards remove the fleshy parts of the bird, and like you keep of the cadaver, and then you keep the yellow part. You can also leave it in the specimen, and then try to dissect around. So that's all the things that we tried, and then and in the meantime we started off what we call soft plastination. That is a relatively new technique in which the starting point is the same as with plastination. So you fix them, fixate them with formaldehyde, and then you dehydrate them with acetone, but then the remainder is completely different. And what you end up with is a soft specimen that is you can keep just on shelves and not in water or in boxes or whatever. And it's you can still move the body parts. And by using that, we saw quite some nice results. May I have the next slide, please? And the problem with these things is that it's so difficult to get it on a picture, because now you see this specimen and you think, yeah, right. Is this what it is? Are you so happy with this one? And yes, we are so happy with this one. I must admit, the the air sac on the abdominal air sac is open, has been opened up here. So what you see here is a young chicken. On the left side is the neck and would have been the head. On the right side is the tail. Downwards you see the leg, and upwards at the left side, left top side, you see the wing. And if you would follow, maybe you can help me, Liedewij, to the grayish part, just on top of the leg. Yeah, to the right, please. A little bit to the right. Yeah, there, this grayish part where you see, no, to the left. Maybe you're there and my computer very slow. Yeah, there. This is the area of the abdominal air sac, and you see the wall of the abdominal air sac. And we opened up this air sac so you can really see through. But this is not so transparent as it was in, yes, thank you, as it was in the alive animal. But it's much more transparent than in any way that we had before. And by opening up the air sac and looking in the bird, you can actually see his genital organs and its kidney, and and go to the cranial side also the lungs. So we're really pleased with this, and we're still improving this technique to see whether or not we can leave this closed and see what we can do then, or maybe we can use an endoscope and show the endoscopic view for the master students, etc. So now with we're still busy on creating, on making these kind of specimens and improving the transparency of the air sac, but still we have it quite, yeah, as I said, we're quite happy with it. So we're trying to I'm looking for a word which I can't find, to have a larger number of birds for teaching. And one of the advantages of this crisis is that I know now that when we are doing the dissection classes for this chicken practical, we can only work with 25 students at the same time, meaning that the number of chicken that we have now for this type of teaching is enough. And that we decided to, already this year, and I didn't expect it to happen, to be honest, but already this year we won't do the dissection class for our first year students, but we gave them, we will give them all types of specimens with the air sacs in it, so also the latex filled ones, etc., but also these ones, and then to see whether or not they get a grip on the 3D anatomy in this bird. So in short, it's already this year we won't be using the fresh chickens for our classes. And see what I had on the last slide. You can go further, please. So in conclusion, it is possible to keep the wall, I should say, of the air sacs more or less transparent, at least transparent enough for our goals. We are using the soft plastination technique. The advantage of it is that the liquids and all the stuff that you should buy for it are much cheaper than with the normal plastination, which gives a hard specimen, firm specimen that you can't bend, that you can't move around. The only problem is because it is soft and it is relatively new. You saw that it is from 2016 and we've been using it since 2 years now. We don't know how sustainable it is in our normal teaching situation. If we can let the students explore everything themselves, or that we should more or less demonstrate it, or it should be demonstrated by the teachers to because I don't know how much wear, how easy it wears or it tears or whatever, and that you have to remake them again and again. So initially it will save, let's say 100 birds a year starting from this year. Maybe we have to replace 1 or 2 every year. I don't know yet. I hope it's not that much. Otherwise we have to change it into demonstration. And we will also are really interested in what the students will say about this, because we know that Over the years, the students are getting more problems with this section of chicken or fresh material. They're always very curious where they come from, and etc. And what I see from the evaluation is that they, well, over the years, they are becoming more conscious of using animals in teaching. And so I'm really curious to know whether they think this is also a good alternative to the original dissection. And that's my presentation. Thank you.
Thank you, Claudia, very much for this very clear presentation in difficult circumstances. Are there any questions at the moment? Oh, at the moment, I can't hear anything. Oh, yeah. Ivo, please.
Yes, Claudia. What is? Are there any plans of making such models more available to other studies like biology, which have comparative anatomy in their curriculum, to ensure that they can give more 3D insight in animals without them having to dissect animals?
Yes, of course. We have been in contact, not about birds, but we have been in contact with people from biology. It's always possible for studies to borrow them from us, but not in large amounts. And they can also ask us to make them for them for cost price. So that's always a possibility to do. And also we can cooperate and give combined classes as well. Yes.
Thank you.
Kirsten, is your hand still up, or do you have a new question?
Sorry, no, it was still up.
Okay, I thought so. Yes. Well, then I have a question. I think these air sacs are a bit the same for all bird species, but could you think of any applications for other parts of other types of animals?
Yeah, well, especially because what we did, of course, is of what we do in teaching is when you look at abdominal anatomy, so belly anatomy, then basically at the moment we use wet specimens. So they're fixed fixated in formaldehyde, and we keep them also in these large trunks of formaldehyde. But we're also trying to replace that one because formaldehyde is bad for your health, and we need to replace them as well. And until recent, it was really difficult to replace them by silicons, the real silicon plastination, because then you get rigid, firm specimen, and then it's almost impossible to move the intestines, and that's what you want if you're teaching an abdominal anatomy, or if the students are studying abdominal anatomy. They have to be in a possibility to move the intestines to see that the kidneys are underneath it, or that the bladder is there, etc. So also with this technique, and then it's not the transparency we're looking for, but it's just the softness. We can create specimens that are movable for the students. And what we're also doing, and that's another funding that we had from this fund, is trying to create a model in which you can insert things so that you can practice, in our case, a vaginoscope. So you can put in a scope as in a practice that you would do as a veterinary treatment or veterinary diagnostics. So by keeping the specimens soft and nearly as soft as would have been in real life, you could try to simulate things as a procedure, and you don't need the live animal for that to practice it. So that's why this technique has also been used for, and we try to use for, and we are now starting, for instance, to simulate the urine taking of a cow and see whether or not we can do the catheterization in a part of the back of a cow. Instead of using the live cows, we can do that in the model, starting with a part that is fixed in formaldehyde but kept soft and yeah, so that you actually can really manipulate the tissue.
I see. Okay, thank you. Well, then we've come to the end of this meeting. Thank you all for joining us. For us, it was a nice experiment to present a few projects which were granted by the 3R Stimulus Fund. It is a fund exclusively for employees and students of the Utrecht organizations, the university and the medical center. The deadline for application is 30 August this year. Thanks to both speakers and thank you for joining us, and keep safe. Bye.
Bye, thank you.